panel of examiners here. My name is Daniel Oro. I'm the president of the panel just for a couple of hours. I'm a professor of research at the Center of Advanced Studies in Blanes. It's a branch of the SIC, the IFISC. Yeah, the disclaimer, I'm not a physicist, I'm a biologist. Okay. Hopefully. Yeah, then to my right, Saul Ares is a tenor scientist also at the SIC, at the National Center of Biotechnology. Right? To my left, Carmen Miguel is full professor at the Faculty of Physics at the University of Barcelona. And we are here as examiners to examine Alex Jimenez to become, hopefully, a PhD. Right?
Yeah.
Okay, Alex, the floor is yours. Yeah, please, please, this guy, who can mute if possible. We'll make a very short break after the oral presentation before the question, right? Just you to know, in case you want to leave the room or whatever, go to the restroom or whatever. Okay, Alex, please.
Okay, thank you for the presentation. So this is my thesis, and I would like to start with the motivation, first of all, for doing this thesis, which is the global biodiversity crisis. This concept is quite well illustrated in this image, where in red color, you can see land habitats in which more than 50% of the original biodiversity has been lost. And the thing is that since the 1970s, natural populations have been experiencing a steady decline. For instance, 10 million hectares of forests have been lost every year since then, or the global vertebrate population has declined by an astonishing factor of around 70%. Indeed, it is estimated that current extinction rates is about 100 to 1,000 times higher than the natural background rate, and between 10 to 50% of the species are nowadays endangered. We have similar examples for marine life in particular. For instance, 50% of the coral reefs have already disappeared, and marine populations have experienced also huge declines. I think this is enough motivation to jump to study these topics. And the first question that one seeks to answer is, which are the drivers or the causes of these biodiversity losses that we are observing? So the main drivers, there are many, but the most important ones were identified in the Millennium Ecosystem Assessment in 2005. And sadly, most of them are from anthropogenic origin. We have habitat loss or change, overexploitation of resources, the establishment of invasive species, climate change, ocean acidification, and emerging diseases. These are the most important drivers, and indeed, all of them are interconnected. For instance, climate change can favor the establishment of invasive species, which in turn can produce emerging diseases, or climate change contributes to ocean acidification, for instance. In this thesis, we are going to study these drivers of biodiversity loss, ocean acidification, climate change, and most importantly, emerging diseases. And we will do so from a perspective of complex systems using mathematical and data-driven models. So I will divide the presentation in three main parts. In the first two parts, I will talk about our results on emerging diseases. In particular, parasite-produced marine diseases and vector-borne plant diseases, using as case studies the mass mortality event of Pinna nobilis and the diseases caused by Xylella fastidiosa. That, of course, I will introduce later. And for the third and final part, we will turn our attention to the monitoring of coastal ecosystems, using as case studies the monitoring of ocean acidification, seagrass meadows, and coral reefs. So these emerging diseases are going to be studied primarily using mathematical models. And for the case of the diseases caused by Xylella fastidiosa, we will combine these mathematical models with data, with climate data, to develop a data-driven model to study the biogeography of these diseases. And finally, for the last part, we will rely on purely data-driven methods such as artificial intelligence or data analysis. Okay, so let's start with the first part. In this image here, you can see a healthy individual of Pinna nobilis, a giant pen shell endemic of the Mediterranean Sea. It can grow up to 1 meter and live up to 50 years. But if you were lucky to spot one of these nowadays, you would most probably see something like this. This is a photo that I took indeed myself about 2 years ago in the Bay of Poyensa, and you can see several individuals of Pinna nobilis that are dead. This situation has been quite common since the year 2016, in which a mass mortality event spread from the west to the east of the Mediterranean in just a few years. As you can see, most of the populations experienced very high mortalities, above 95%, but you still can see that in some regions, they were kind of protected. So it was later found that this Hoplosporidium pinne, this parasite, which is novel to the Mediterranean Sea, is the responsible for the mass mortality event. So in the end, the mass mortality event is an epidemic. It was also shown later that this is spread correlates with marine currents. So it is thought that the parasite is passively transported through these currents, and that disease expression depends on the environmental conditions. For instance, there is a threshold, a temperature threshold, below which we don't have disease expression, below this 13.5 degrees. And this disease expression only occurs in a given range of salinity. And indeed, this salinity dependence is what makes that these regions in which the disease was not found, basically. Okay, so despite all this information that was gathered kind of quickly after the mass mortality event, there were no efforts to try to model the disease, to try to understand it further, or try to predict its future expansion. And the thing is that modeling parasite-produced marine disease is not straightforward. Indeed, at the beginning of the century, it was not even clear if the tools or methods from terrestrial epidemiology could be used to marine systems. And we can see that compartmental models, which are canonical in terrestrial epidemiology, are just starting to be used, as you can see, for instance, from this reference. However, these models that are being used are too complex, so they depend on many variables and parameters, and so they require a lot of data to be fitted, as you can see here. This is not useful for emerging diseases because by definition, we don't have much data. And there is a second drawback because of the complexity of these models. We cannot perform detailed mathematical analysis, and this in the end prevents our deeper understanding of the processes that we are modeling. So to try to overcome these limitations, we developed the simplest model we could think of for parasite-produced marine diseases in sessile, so immobile hosts, keeping in mind an application for the mass mortality event of Pinna nobilis. In our model, we assume that the host can be in one of three states, susceptible to the disease, infected, or dead, and that parasites are free in the medium. The idea is that susceptible hosts absorb the parasites and get infected at a given rate beta. Then infected individuals produce and release parasites to the medium at a given rate lambda, and also die at a given rate gamma. And parasites, as I said, are free in the medium, can infect susceptible hosts when they are absorbed, and also die at a given rate mu. So assuming that the system is homogeneous, well mixed, basically that any parasite can reach any host at any time, we can write this system of differential equations that will describe the dynamics of the system. Now, one of the most important things in epidemiology is the basic reproductive number, R0, which provides a number of secondary infections given by a primary infection in a fully susceptible population. This is the typical or basic definition of R0. But it is so important because this quantity defines the threshold for the occurrence of an epidemic. So if this number is below 1, there won't be an outbreak, while if it is greater than 1, an epidemic will occur. So to find an expression for the basic reproductive number, one of the methods to do so is, well, first we have to find the disease-free state of the system. We have to check that this disease-free state is a disease-free equilibrium or fixed point of the system of differential equations. And then, for instance, we can perform linear stability analysis to obtain an expression for R0. We can also use standard methods related to this, which, for instance, the next generation matrix method. And with these methods, applying these methods to this model, we are able to obtain this expression for the basic reproductive number. Now, as you can see, this expression here depends on the 4 parameters of our model. So to know a little bit more about the mass mortality event of Pinna nobilis, we would like, for instance, to know R0. And for that, we need to know the values of the parameters. To do so, you usually feed the model to data. In our case, the data that we have comes from a field conservation effort that was done in Portugal, in Catalonia. So there was a natural population of pen shells here that they thought that was not affected by the disease. So they collected them and put them in some water tanks at constant temperatures. What happened later is that an epidemic developed in those water tanks. So the disease was already there. At least we can use this data to fit our model. But as you can see, the only thing that we have is the number of dead individuals in time, nothing more. For instance, we don't know anything about the dynamics of the parasite population. So we cannot fit our model because we have an equation for P. So we have to try to reduce it, basically using mathematical analysis. This is what we did next. We show that our model has a conserved quantity, this one, that allows us to find a close relation for the dynamics of the parasite population as function of the dynamics of the states of the host, which is this one. And this in turn allows us to reduce our model to have a simpler model that not longer depends on P, on the parasite population. But still, we can see that this still depends on 4 parameters. So it's going to be difficult to fit this model to the data that we have. Then we discuss an approximation. Under this condition, we can get rid of this term. And then this simplifies the subsequent equations and the reduced model that now is not exact, is approximate. But still, it still depends on 4 parameters. But we can see that this beta parameter is multiplying both lambda and the initial condition. So we can redefine these products to a new parameter, a new initial condition, and then the model depends only on 3 parameters. Okay, so this model will be easier to fit to data. Of course, we can validate all these analytical findings that we have obtained. Here in this image, in solid lines, you can see the numerical solution of the original model of the 4D equations. Then in dashed lines, you can see the numerical solution of the exact reduction of the model. That, of course, it's in perfect agreement at all times because it's an exact reduction. And in dotted line, we show the approximate model. You can see that in panel A, where we are not fulfilling these conditions, of course, the approximate model does not describe well the dynamics of the system, but in panel C, as long as we fulfill these equations, the description of the dynamics is perfect by the approximate model. We can even go further. Here we discuss a timescale approximation in which if parasites die faster than infected hosts, then the dynamics of P can be approximated by this equation that allows us to obtain this effective SIR model. For the ones that don't know what the SIR model is, this is like the typical, most famous and simple model in epidemiology. So we know a lot about this model. And we see that we can use it to describe the mass mortality event of Pinna nobilis, for instance, if this situation is fulfilled, of course. Again, we can show that this reduction works if these conditions are fulfilled. This is what this image shows. And finally, after all this mathematical analysis, we are able to, or we try to fit the model. First, we start with our most complex model. We try to fit it to data, and we find that we are not able to find a unique combination of the parameter values that fit the data. Instead, we found some relations of 2 of the parameters of the model. So if these ratios are fulfilled, we can fit the data perfectly. And indeed, we have a problem of parameter unidentifiability, but if we look closer, these ratios are nothing but the effective transmission rate of our effective SIR model. So what if we directly fit the SIR model? And then, of course, we obtain that we can fit the model. We obtain a unique combination of the parameter values that fit the model. And something interesting here is that even if this model is very simple, it's just the SIR model, we recover something that we know from the disease, which is the dependence with temperature. So for the tank that was kept at higher temperature, we have a higher transmission rate. So it's interesting. Now, all these results come from a model in which we assumed some homogeneity condition, and we didn't study how the mobility of the parasites is affecting the disease dynamics. So we decided to extend our model to take this into account. Here we define a grid in which the hosts are placed. Remember that the host cannot move, and the parasites will move through this grid. All the other processes are the same as before. So we have a new parameter, the mobility of the parasite. Now, to solve this system, we consider an individual-based model, and we solve it using the Gillespie algorithm. So this is a single realization of this model, which is stochastic. So each of the realizations will be different. And this is what you see when you simulate it, basically. So let's see which results can we obtain from this model. First of all, we see that the high mobility limit, so when the parasites die and move many times before dying, which is panel C, our system of differential equations that we had previously is able to describe the spatial model. So the system of differential equations is the limiting case of the high mobility regime of a spatial model. Of course, if you are far from this limit, then the system of differential equations does not describe the dynamics of the spatial model. And next, we obtained an expression for the spatial basic reproductive number. Here, there is no exact method to obtain this. There's no method to obtain an exact relation, but we came up with an approximation, which is this one here, and we showed that it is able to differentiate, to separate the disease-free phase from the propagation phase, which is the property that you want for R0. Now, it's interesting to see that this spatial R0 is a product of the non-spatial basic reproductive number and a mobility factor. So the main ingredients of the biology of the disease would be in the non-spatial factor. And then we have a mobility factor. So these two things are uncoupled. And if we go to the high mobility limit, in which kappa is higher than mu, this goes to 1, and we recover the basic reproductive number of the non-spatial model. So not only the differential equations describe well the dynamics, but with this expression, we also recover it. So it's quite interesting, at least from the theoretical point of view. Of course, we can compute or study more things. Here we show that the disease spreading velocity of the infected population can be related to the parasite mobility rate and also to the value of the non-spatial basic reproductive number. So from these two quantities, we can predict the disease spreading velocity. This was similarly done in previous works. And we also showed that the time to extinction can be related, again, to the parasite mobility rate, at least for high values of the spatial basic reproductive number, in which we can obtain this formula. That fits very well. So these last results are indeed quite important from the perspective of policymaking or to develop strategies of control for this disease or for predictions, for instance. So with this, we would finish the first part. Let's see the results about emerging diseases, this time vector-borne plant diseases, exemplified by the diseases caused by Xylella fastidiosa. So let me introduce Xylella fastidiosa. Xylella fastidiosa is a very dangerous plant pathogen that can affect many plants. It has a wide host range, can affect up to 600 different plant species, including important crops, such as the one in the image, for instance, almond or olive trees, citrus, and vineyards. So as you might imagine, this can cause huge economic impacts. For instance, in California, where Xylella fastidiosa causes Pierce disease in grapevines, it causes $100 million of losses every year. These diseases are transmitted by xylem-feeding insects, and they were originally, the disease was originally restricted to the American continent, but it has recently spread to other parts. For instance, since 2013, it was introduced in Europe and also to other places in the world, as you can see here. And now it is confined in the Mediterranean region. We can find it in the Mediterranean region, mainly in Europe. Okay. So these diseases are quite complex. We have the disease cycle, which includes a vector now. So the vector has its own life cycle. For instance, for this case, for these diseases, only the adult stage of the vector is able to transmit the disease. And because of this life cycle, we can find these abundance patterns of the adult population that can be observed in the field. Of course, on top of all that, we have environmental conditions such as climate, temperature, for instance, that influences all these processes. So let's see which is the background to model these complex diseases. But we find that previous model basically lacks realistic descriptions of some of the underlying processes. For instance, until very recently, the vector population was not modeled, and the vector population is the responsible for disease transmission. And yet, the seasonal abundance patterns are not included in the modeling. Of course, all models are wrong, but some are useful. I think that all of them are useful in a sense, but we want to include the vector seasonal abundance patterns into the modeling to see how this affects disease dynamics, and also to add a little bit more of realism. So indeed, we see that considering an exponential decay, we can fit pretty well these abundance patterns. So the first thing that we did was to consider a simple model that would represent one of these decays only, in which basically we want to know how an exponential decay affects disease dynamics. So we have a vector-borne disease model in which the initial condition of the vector population decays until reaching an equilibrium value. Of course, if your initial condition is below the equilibrium value, it will increase. But we are mostly interested in this part. Anyway, we study both. So the difficulties here came when we wanted to compute the basic reproductive number, R0, because basically if you compute the disease-free state and you check if it's a fixed point of the system, you see that it's not by definition because the initial condition is out of equilibrium. So we didn't know how to compute R0. We looked at the literature and we see that some researchers were invoking some theorem and they were able to compute R0 from the post-pandemic fixed point. So basically in our system, this would be when the vector population reaches the equilibrium value. Of course, there you do have a fixed point and you can apply the standard methods. This is what some people were doing. So we applied same method. We can compute, of course, the equilibrium value of the vector population, plug it in here. Now we have a fixed point. Now we use linear stability analysis or next generation matrix method, and we obtain this expression for R0. However, we observed later that this is not working, at least not always. For instance, if you look at this part of the plot, we can see that in some cases, when R0 is smaller than 1, there are some big outbreaks in which 80–90% of the population dies. So this is not working. In other cases, this works. Indeed, all this is explained in the thesis in detail. We can show mathematically why it works in each of the cases. But the important thing is that for some of them, this was not working. So we propose an approximate solution to solve this issue. We define an instantaneous basic reproductive number, which is basically the previous relation that we had, but substituting the vector population at equilibrium by vector population at time t. And we propose that the basic reproductive number is the average of the instantaneous value over one generation. So basically this relation here, and by applying this to our model, we obtain this expression for the basic reproductive number in this out of equilibrium situation. It's a little bit ugly, but it works quite well. So at least with this definition of R0, or with this expression, when R0 is, there is only an outbreak when R0 is greater than 1. That this is the behavior that you want for R0. So other things that we did here was to discuss some reductions of the model that are useful for emerging diseases. I just want to highlight this one. Again, if we consider a timescale approximation in which the vectors die faster than infected hosts, we can reduce our model to an effective SIR model again. So it's a nice parallelism with the marine diseases. Using the same kind of approximation, we obtain the simple SIR model. And this is going to be important later for another chapter of the thesis. Okay, so once we solved this issue with R0, we were ready to develop a model for Xylella fastidiosa diseases that took into account the vector seasonal dynamics. Now, not only 1 year, let's say, but all the patterns. We did this and show using Bayesian inference that we can fit some data available for 2 different outbreaks of Xylella fastidiosa diseases. One for almond leaf scourge disease in Mallorca that affects almonds, and another one for olive quick decline syndrome in Puglia that affects olive trees. Another interesting thing here is that we see that with our definition of the basic reproductive number, the value R0 equal 1 is obtained close to the peak of the epidemic, which is, again, the expected behavior of R0. So it seems that our method is working pretty well. Now, why it is important to take into account the vector seasonal abundance patterns? Maybe we are overcomplicating our life, and maybe we are, but we find that if you perform a sensitivity analysis to the model, you can see that for important epidemiological quantities, this parameter mu, which is the one that regulates the abundance patterns, is one of the most important parameters of the model. So it seems it's important. And also, secondly, taking into account modeling these vector abundance patterns we can design or propose new control strategies. For instance, something that is done nowadays to try to control, to some extent, these epidemics, is to try to lower the density of vectors, the initial density of vectors in the fields. However, we see that this is far from optimal, but if we could reduce the period of time the vectors are in the field, basically our parameter mu, this would be more effective. And indeed, the optimal strategy is a combination of those. So it would be nice to think on how to do this in practice, which for sure won't be easy, but we can think about it. So if you remember, I said that these diseases were originally restricted in the American continent, but now have jumped to, for instance, to Europe. In particular, in Europe, there is a lot of concern about whether or not these diseases can establish and spread throughout Europe, because of course this would have huge economic impact. So here in this thesis, we also wanted to answer this question. Now, we cannot use our previous developed models, these compartmental models that you have seen that they depend on some parameters, and the values of those parameters depend on the environmental conditions. So for each of the combinations of environmental conditions in Europe, we have different values of the parameters. So it is impractical to obtain all these values, even if we had the data, but in addition, we don't have the data. So we have to find another method to do that. And people has been using to approach these problems, basically species distribution models, in particular correlative species distribution models, which are usually machine learning models that use presence data of a given species that you want to model and correlate this presence data with climatic variables to finally predict the probability of presence of this species as function of the environmental variables. This probability of presence is also called usually suitability. So for the case of Xylella fastidiosa diseases, some suitability maps were developed for the pathogen Xylella fastidiosa. And for the vector, in the case of Europe, we have basically one main or only relevant vector, which is Philenops fumarius, and this is the suitability map of this vector. So of course, this provides a lot of information. However, this is not telling us nothing about disease risk, because a disease is the emergent phenomenon of the interaction of the pathogen, the vector, the host, influenced by the climate, etc. So with these two informations independently, we cannot really say which is the risk of having the disease established in Europe. So to try to answer this question, we developed a data-driven model. Now for Pierce disease, which is a Xylella fastidiosa disease that affects grapevines. Why Pierce disease? Because the data that we needed to build this model was only available for Pierce disease. But in general, you could extend it to the others. So basically, we consider two main processes to model. The first one is the transmission of the pathogen from infected to susceptible hosts, creating exposed hosts. This is a process that is modulated by infected vectors, of course. And then there is a second process that we model, which is the development of the infection from these exposed hosts. It means hosts that have just acquired the bacterium, basically. And this process depends on the growth and survival processes of the pathogen population inside the plant that will depend basically on temperature. This whole thing happens on an annual basis. We can divide it in two main seasons: the growing season, in which temperatures are warm enough for the pathogen population to grow and to produce the infection in the host. And then we have also the curing season, in which if temperatures are cold enough, the bacterial population can die and then the plant recovers. Here, recovers mean that they are susceptible again because there is no immunity in this disease. So let's see how we model this. First, we use our previous vector-borne disease model, and indeed, we make use of the timescale approximation, which is suitable here in this problem. And we even simplify this further. We use the initial phase approximation of the desired model, which basically tells you that initially, the infected population grows exponentially at a given rate proportional to the basic reproductive number. Now, because we started with this vector-borne disease model, we know how R0, which is basically our only free parameter, because gamma is known, depends on the original values of the original parameters of the vector-borne disease model. For instance, we are going to make use that R0 depends linearly on the vector abundance, because vector suitability is a proxy for vector abundance. So in this case, we can obtain, we can use a spatially dependent basic reproductive number in Europe to make later our predictions. So to model the development of the disease in these exposed hosts, we use some available data on the Xylella fastidiosa growth rate that depends on temperature, as you can see here. We feed this data, and with this feed, we define a metric of temperature accumulation, which we call modified growing degree days, which is basically this expression here. And we show that this metric can be related to the bacterial load accumulated inside the plant after a given period. So basically, if we consider, for instance, a logistic model for pathogen growth, but we take into account that the growth rate of the pathogen depends on temperature, that in turn depends on time, and we integrate this, basically, we obtain this expression, and we see that this depends on this integral here, which is basically our definition of modified growing degree days. This F, well, this is the growth rate, which is this. So this is also the approximated version of the integral, but it's the same thing. So the idea is that the more bacterial load a plant has, the more likely it is that it develops infection and it shows symptoms. So here, two biologists that were collaborating in this work, Eduardo Moralejo and Marina Montesinos, performed these nice experiments in which they inoculated the plants with the bacterium. They tracked symptoms over time and also recorded temperature. So with this, we could compute the modified growing degree days during this time interval and relate it to the probability of developing the infection. So basically performing this fit here. Now to model the winter season, the curing season, basically we don't have the same data that we had before. We would like to have the death rate of the pathogen as function of temperature and then perform the experiments, but we don't have any of this. So we have to rely on previous work. In this way, all the details are, of course, in the thesis. We can obtain this look curve here that relates the cold accumulation with the probability of recovery. Here I'm showing the inverse of that, which is the probability of keeping infected after winter as function of the accumulated cold. So joining all this together, we can build the final model. So here, basically, we have the transmission part. This part tells us from a given previous condition of infected individuals, the number of new exposed individuals or exposed individuals for that year. Then we have the probability of infection, which is the proportion of those that will get indeed infected for the next year. This comes from two terms. First, the development of the infection during the growing season, and then which of those will keep this infection after winter. So this is what the model does. You iterate this and you can simulate the disease dynamics. And then we can define a risk index as the effective growth relative to the maximum theoretical one. So this is the maximum possible growth under optimal conditions when the probability of infection is 1, basically. And this is the realized growth. So if this index is negative, we have an exponential decay of the infected population, so there is no risk there, basically. If it is positive, we do have risk. And we can even try to define different, we called them risk zones, but we could say better impact zones, because we correlate the positive part of the risk index with the potential impact. So once we have developed the model, what we did is to validate it using spatiotemporal data for the presence and absence of Pierce disease in the United States, where the disease is fully established. And we can obtain R08 for the free parameter of our model. And we see that with that choice, we obtain overall very good accuracy. So once the model is validated, we can finally apply it to Europe. That is what we wanted. So here we use, as I said, a spatially dependent basic reproductive number using the suitability of Philenosis spumarius, which is this map here. And finally, we obtain our results. This is the risk for having Pierce disease outbreaks in Europe. And we basically see, as first sight, that the risk is confined into the Mediterranean region. Something interesting is that we see that in all places in which Xylella fastidiosa diseases in general, not only Pierce disease, has been found, our model is saying that there is risk there. And something interesting as well is that by the time that we published the paper, Pierce disease was only found had been only found in Mallorca, but afterwards it has been found in Portugal and in this region, indeed this year, I think, in this region of Puglia. So that's quite interesting because it seems that the model is working well. Okay, and of course we did more here. One of the things that were interesting that we obtained was that we observed that the risk was increasing. For instance, in this region of France, you can see it here, the surface at risk has been increasing since the 90s. So it seems that climate change is changing the situation. And then the next question is what is going to happen in the future, which is the risk future. So we decided to study that. Indeed, this is interesting per se, because if you look at the climatic suitability of the pathogen and the vector for different levels of global warming, you can see that global warming is going to promote the growth of the pathogen, but it's going to be detrimental for the vector. In most of the places, the suitability decreases. This was already done in previous work, and the question was, what about risk? So we didn't know what was going to happen, but with our model, indeed, it's straightforward because we just have to run our model and check the results. What we obtain is that in general, the risk increases, but most importantly, we found that at this +3 warming level, it's a kind of a critical point in which the disease could be established throughout continental Europe. But before, it is still confined into the Mediterranean region. Of course, here we studied more things. We developed basically indeed a web page to show all our results, both for the risk of disease in Europe, both in current and future climate. We show, for instance, the risk at different warming scenarios at the country level, or protected designation of origins, or also at the vineyard level. So if anyone is interested, there is this web page you can access through that link and check these results. But still, we had one more question about this, which was which is the effect of the spatial resolution into our predictions, the spatial resolution of the climate data that we are using. Until that point, we were using data from ERA 5, in which each pixel is a 10 by 10, more or less, kilometer pixel. But what if we used higher resolution climate data, for instance, from CHELSA, in which each pixel is a 1 by 1 kilometer pixel? Looking at this picture, for instance, the maximum daily temperature of a given random day, it seems there is not that much difference. Basically, you can see better the spatial patterns here, but it seems it's kind of the same. But when we computed the risk, we saw things like this. With ERA 5, with lower resolution climate data, if we look at Galicia, for instance, and the north of Portugal, it seems there is no risk. But using higher resolution climate data, we can see that some specific patterns emerge. For instance, Minion River, and more rivers and valleys emerge as zones with risk. So if you think about it, in these rivers and valleys where vineyards are usually planted, so this is especially important for peach disease. So we wanted to study this in more depth, and this is what we did. So we show that indeed there is a global risk upsurge due to a higher resolution climate data. So there is a lot of difference, basically. And this difference in risk index is quite important because it changes the risk categories. For instance, there are a lot of zones that change from no risk to risk, or that have a risk category increase. Some of them also have a decrease, but most of them have an increase. And as I said, this is especially important in rivers or valleys. For instance, in this plot, you can see in black dots known locations of vines, or vineyards. And you can see that few of them fall in these places in which the risk changes. So if you look at this in more detail and you correlate the risk changes into these locations, you can see that there is a lot of change. And indeed, the risk categories changes. A lot of points change from no risk to risk or have a risk category increase. If you look at the distribution, you can see it very clearly. So the distribution is shifted to the right, so to a higher risk. So all these results are quite important for policymaking, because if you want to know the risk at the vineyard level, you should use high resolution climate data. Of course, that's not easy, but we should try to do it. Okay, so with this, we finished the first two parts. And I will now show some results about the monitoring of coastal ecosystems. I will do a summary. I won't explain this in detail, but the most important things. So the first case study is about measuring or monitoring ocean acidification at the local level. So here you can see a known trend of ocean acidification at the global level. This can be measured, and then you can obtain this trend. However, to measure this locally is a way more challenging, usually due to data gaps. For instance, here I'm using some data of 2 stations. I'm showing temperature, salinity, and dissolved oxygen in these 2 stations of the Balearic Islands, Palma Bay, and Cabrera. And you can see that there are a lot of data gaps in this time series. And this is even worse for the pH measures. For instance, since 2013 until 2019, there's no data. And after that, there are still some gaps. So we thought about developing a machine learning model to be able to reconstruct this time series, to then be able to measure ocean acidification. Here we use a long short-term memory neural network, which is quite suitable for time series forecasting. The idea is that we want to predict a given central part of the pH time series using past and future information with respect to that point, of course, of the other variables that are available, temperature, salinity, and oxygen. This is the idea. And well, we show that the method works pretty well. We are able to train the model. The predicted values are in good agreement with the true values, as you can see more visually here. And then this allows us to fill these gaps, which makes sense as well. And for the case of Palma Bay, we can even reconstruct this long time series. Of course, this reconstruction is not perfect for sure, but it's an estimation. And using this complete kind of time series, we can now obtain the ocean acidification trend. And the number that we obtain indeed is in very good agreement with other regional measurements that had been done in, for instance, for France. So it seems that the method is working quite well. The next case study. Bueno. Así puedo ver. Así. Ya está, ¿no? Okay. Well, the next case study is about the monitoring of grass meadows, in particular Posidonia oceanica in the Mediterranean Sea. These ecosystems are quite threatened nowadays due to global change, mainly pollution, but also global warming. There is a known temperature threshold, which is 28 degrees, above which these plants start to die. Indeed, over the last 100 years, we have already lost about 20% of the seagrasses globally. So it is quite important to monitor these ecosystems. However, again, this is quite challenging. So we can either rely on particular measures, basically field sampling, that cannot cover a wide extension, or if they do, it would be with sparse data. Or we can rely on other techniques, for instance, side scan sonar, that it's typically equipped in ships. And with this technique, you can cover wide areas, and you have very, very good data, very precise data. But this is very expensive and time consuming. So it is the balance of these 2 things. But now there is an emerging possibility, which is using deep learning methods or machine learning methods to predict or obtain the benthic habitats from satellite imagery. This idea is not new. It has been previously done. There are some previous work which basically constitute a proof of concept for that methodology. But now the challenging is to develop a model that it is useful, that can be deployed on a real case scenario. This is the real challenging now. And this means that we are able to train a model that it is able to obtain the benthic habitats, in particular of Posidonia oceanica, beyond its region of training. Because what it's usually done is you have some data of a given bay, you train with the scarce data you have, you predict there. So that's okay, but it's not very useful. You cannot go to the bay next to this one and predict because it will fail. Indeed, we found it in our study as well. So here what we did to push on that direction is to develop a deep learning model that is based on a consensus of 10 convolutional neural networks, which are quite suitable for image segmentation problems. And we basically take advantage of this huge availability of data that we have for the Balearic Islands, for the benthic habitats of the Balearic Islands. This data comes from side scan sonar, so it's a very good data. And what we are going to do to try to obtain this deep learning model that it's able to generalize to other regions, or to try it, is to train with the data with Mallorca alone. Only with this data we train the model. And then we propose a very challenging test, which is basically predict in the other islands, Menorca, Ibiza, and Formentera. This is very challenging because well, first of all, each of the images that you have from the satellite comes from a given angle, from a different position respective to the sun, to the satellite, etc. All this changes continuously. Also, the bottom types in the test set are different from the ones in the training set. For instance, the type of sand is different here and here than in Mallorca. And even there are some species that are present in the trained dataset and are not in the test dataset, or the other way around. So all this, of course, complicates a lot that this can work, but we try it. And what we find is that after training the model, of course, for the training dataset, we have super good accuracies. And in the test set, of course, we have not that good accuracies, but still are not that bad. So we are able to predict these test sets with quite good accuracy as well. That's interesting. And then we show that indeed, if you are interested on measuring the extent of, for instance, of Posidonia oceanica, this is for which we are interested, we see that we can predict the true extent of Posidonia oceanica, the green dots, quite well as well. Another thing that we did here is to obtain the typical error that our model is committing, so that afterwards, if we go to another region and we measure, we can at least associate this error as a minimum error that we know that we are committing. So just that to show a little bit more visually how the model is working, this is an example of the results in the training set. This is an image of Poyensa Bay. This is the ground truth obtained with the side scan sonar. And this is the predictions of our model, which are very good. But of course, this is in the training set. But if we go to the test set, for instance, this is the whole benthic habitats in Formentera. Again, this is the satellite image, the ground truth, and this is the prediction, which is really good. And the model has never seen anything about this zone. So these results, I think, are quite promising. But of course, we have to work more on that. So the last thing that we did was to train the model with all available data to obtain the most up-to-date model, the most powerful model we could build. And well, we developed a web page to show all these results. If anyone is interested to see for the different regions, you can see it. And something I think it's important is that we uploaded these models to GitHub. Anyone can download them and use them. Of course, you need the satellite images, but you can download them. And with this, we go to the last case study, which is about the monitoring of coral reefs. So here we didn't do the monitoring. This was already done, but it's an example to show that we can use this data from a large scale monitoring to obtain nice or interesting results about the ecosystem we are monitoring, not just to monitor per se. So the data that we use here come from the Allen Coral Atlas, in which we have benthic habitat data for coral reefs for all tropical coral reefs worldwide. As you can see in this figure, these are divided into different coral provinces. A province is just like a collection of coral reefs. And as you see, they have data for all the world. So what we do here is basically analyze this data, and we find very, very interesting results. We have found some universal scaling laws or macroecological patterns for the structure of coral reefs. For instance, if you measure the size distribution of the coral reefs in all of these provinces, each province is a color, you obtain the same law, a power law with the same exponent. on average, of course. We also measured different metrics, the area perimeter ratio, the inter-reef distance distribution, and in all of them, we see this kind of universality. From this area perimeter relationship, I'm not going to explain it in detail, but you can see that the reefs are fractals or have fractal geometries. We computed the perimeter and surface fractal dimension for these reefs. Again, we see some sort of universality. And something interesting is that if you consider each of the coral provinces as a single object now, and you compute the fractal dimension of that object, again, some sort of universality. So in all the provinces have some fractal dimension, and they have more or less the same fractal dimension. So it seems it's again a universal result. We think that this is, or we hypothesize that this might be because coral reef provinces could be understood as a higher organizational level of coral reefs. But of course, more research is needed to know it with certainty. Okay, so with this, we finish the results of this thesis, and I would like to just conclude briefly. First, to show the main, or highlight the main contributions of the thesis. For marine epidemiology, we have developed a quite general model for marine diseases in sessile hosts. Sessile means immobile. We see that under some conditions, we can reduce, simplify these models, and this is useful for emerging diseases because then we have less parameters to fit. For instance, we see that the spatial model can be approximated by the non-spatial model in this high mobility regime, and in turn, the non-spatial model can be approximated by an effective SIR model under some conditions. Of course, our results are also important for the mass mortality event of Pinna nobilis in particular, to try to, control the epidemic or just to further understand it. About vector-borne plant diseases, the main thing I want to highlight is that we have shown that we have to be careful when computing R0 if the vector population is out of equilibrium. Of course, this would apply to other compartmental models as well. And we have provided an approximate solution to this problem, which seems to work pretty well. Of course, modeling the vector abundance patterns of Xylella fastidiosa diseases in particular, we show that it provides insights for new control strategies and that it's important. We have also shown that. About disease biogeography, well, we modeled the role of environmental factors in the establishment of vector-borne plant diseases. This was not done. We couldn't do this with species distribution models, but now we can do it. We have obtained, using this model, the risk of establishment of Pierce disease under current and future climate conditions. And we have shown that it is crucial to consider high-resolution climate data for policymaking. For the monitoring of coastal ecosystems, we have successfully developed different data-driven methods to monitor coastal ecosystems. For instance, the reconstruction of measured pH time series using this long short-term memory neural network in the Balearic Islands has allowed us to estimate the local acidification trend here in the Balearic Islands, in Palma in particular. And the model for mapping the Poseidonian Oceanica meadows, we have seen that in principle could be applied to other regions. This is the idea. This is what we want to do, and it's done. And as a last example here, we have shown that the generated data from these large-scale monitorings can also be used to obtain novel insights about these ecosystems, for instance, in the case of coral reefs, as an example, where we show that we have found these universal scaling laws and fractality. So some ideas for further work, just to finish. In marine epidemiology, I want just to say some very few ideas. Something that is left to do is to understand the mechanisms for the rapid spread of the mass mortality event of Pinna nobilis. This has been very quick, and we don't know why yet. Something that could be done is to extend our model by using a meta-population approach to see if we can answer this question or obtain some insight. About vector-borne plant diseases, something that would be really interesting is to study in detail the mechanism that is producing the outbreaks in this out of equilibrium situation, because usually it's a fixed point that becomes unstable. But in our case, this is not the case because there is no fixed point. So let's say that this mechanism that is producing the outbreak is another one. So it would be nice to further study this. About disease biogeography, you can extend the model to other diseases, or something maybe interesting is to incorporate expected mobility of the vectors or of infected hosts by plant trait networks to try to predict the more immediate risk of having the disease. So these risk predictions that we have are basically the suitability for the disease to spread, but this would tell you the more realistic current risk. And about monitoring coastal ecosystems, something interesting would be to measure the spatiotemporal changes of Posidonia oceanica cover in the Mediterranean Sea using past images, and correlate these results that we would obtain with global warming to study to the extent of the temperature-driven mortality that has occurred or not in the Mediterranean Sea. And about monitoring coastal ecosystems, I think this slide shouldn't be here, so because it's the same as before. So I would finish with the publications. These are the publications inside this thesis. These are some other publications done during the PhD. And with this, thank you for your attention, and I will be happy to answer your questions.
Thank you. The Viva event takes now a very short break. Okay, in case you plan to